Welcome to part 3 of the Central Dogma series. This video will explore RNA and transcription. If you would like to learn more about nucleic acids, DNA, and DNA replication, check out part 1 and 2 of this series. In the previous videos, we talked about the building blocks of genes, DNA and RNA, what they are made of, and how they are built. When it comes to replicating DNA, the first portion of Central Dogma, 4 enzymes are required. Helicase unzips the double-stranded DNA. Primase primes the beginning of the new strand. Then DNA polymerase attaches and starts building the new strands. The DNA polymerase of the leading strand continuously moves from the 5' end to the 3' end. The lagging strand is created in shorter, interrupted portions called Okazaki fragments. There are spaces between the fragments that are incomplete. The ligase glues the fragments together to complete the lagging strand. During DNA replication, all the enzymes are working simultaneously along the DNA strand. DNA replication differs between prokaryotes and eukaryotes. Prokaryotes have one point of origin to begin replication and divide into 2 cells while the DNA is being duplicated. Eukaryotes, however, have multiple points of origin throughout the DNA strand, and this is then followed by cell division or mitosis. In this video, we are going to talk about transcription and RNA, the next process of Central Dogma. RNA stands for ribonucleic acid. RNA is small sections copied from DNA and is responsible for transporting genetic material messages to eventually form proteins. RNA is made up of adenine, cytosine, guanine, and uracil instead of thymine. A phosphodiester bond is made between the sugar and a phosphate, just like DNA. Let's look at that bond formation again. RNA sugars have an additional hydroxyl group at the 2' carbon, while DNA nucleotides have a hydrogen at this position. Here we can see the highlighted groups that must break away to allow this bond to happen. 2 hydrogens and an oxygen are removed to form water, and a phosphodiester bond between the sugar and phosphate group is created. RNAs found in Central Dogma include messenger RNA, which are small molecules that have been transcribed from DNA and will eventually be used to create proteins, which determine flower color and leaf shape and size. Transfer RNA, which are molecules that carry amino acids to the ribosome, and ribosomal RNA. For now, we will focus on mRNA. Transcription refers to the process of copying a segment of DNA to make RNA containing a specific gene. The tools needed for transcription are much simpler than DNA replication. We need a strand of DNA and RNA polymerase. RNA polymerase can attach, separate, copy, and reassemble the DNA. While RNA polymerase is attached to the DNA, it will separate the strands. The RNA polymerase will then move down the DNA strand to make a single strand of RNA. But during this process, how does RNA polymerase know where to start and stop when copying the DNA strand? RNA polymerase starts transcription by interacting with a promoter site on the DNA. Transcription starts down the DNA strand from the promoter site. RNA polymerase continues transcription down the DNA strand until it interacts with specific terminator sequences on the DNA strand. This stops transcription, and certain proteins signal RNA polymerases released from the DNA strand. Now we have a strand of copied RNA that we can refer to as messenger RNA or mRNA. The completed mRNA strand will contain a site for a ribosome to bind, a start codon to begin translation, and a stop codon to end translation. It must be delivered to a ribosome located in the cytoplasm of a cell where it will be synthesized into a protein. In a prokaryote, mRNA is synthesized and released directly into the cytoplasm and is delivered to a ribosome immediately for protein synthesis. In a eukaryote, mRNA is made in the nucleus and must be delivered to the ribosomes in the cytoplasm. However, transport through the nuclear membrane is regulated so the mRNA must be manipulated to pass into the cytoplasm. Although the mRNA in the nucleus is a copy of a small portion of DNA, it still needs to be processed before it is transported into the cytoplasm. If we look at the different parts of RNA, it is made of introns and exons. Introns are a portion of mRNA that do not code for amino acids. Exons are segments of mRNA containing information coding for a protein or peptide sequence. To begin the maturation of mRNA, a 5' guanosine cap is attached at the 5' end, and a poly A tail is attached at the 3' end. Then the introns are spliced out to form a mature mRNA. Now the mRNA can be exported through the nuclear membrane and will be delivered to the ribosomes in the cytoplasm for protein synthesis, which will be discussed in the next video. Let's review what we have learned. RNA polymerase attaches to, separates, copies, and reassembles DNA to create RNA. RNA polymerase starts and stops transcription by attaching to a promoter site, reading a segment of DNA, interacting with a terminator site, and releasing from the DNA. The end result is a molecule of mRNA. In prokaryotes, mRNA is made directly from the nucleoid in the cytoplasm and delivered to the ribosomes for protein synthesis. In eukaryotes, mRNA must be modified by removing portions called introns and adding a 5' guanosine cap and a poly A tail in order for it to leave the nucleus and be delivered to the ribosomes. RNA's principal role in central dogma is to carry instructions from DNA for the synthesis of proteins for organisms to perform biological functions.